Our group selected the Inheritance Radio Lab as a basis for our experiment. We wanted to continue on with the idea that you can genetically alter someone else's makeup.
DNA gene splicing is the practice of taking parts of DNA from one organism and adding them to another. Using this method, one may be able to isolate or even combat certain viruses that are passed down from parents to their offspring.
The animal in the experiment is the marsh rice rat, which also can carry a disease called the Hantavirus Pulmonary Syndrome, which can be passed on to humans. We also found it interesting that it was more concentrated in the West where we live.
Hantavirus Pulmonary Syndrome, or HPS, is a deadly virus to humans that is passed to them through rice rats. There is no cure or vaccine for HPS. So what if DNA splicing was involved? Could the virus be exterminated at their point of origin? Our idea is to pinpoint the unhealthy gene within a rice rat and replace it with another rat's healthy gene. This would prevent the carrying on or passing of the bad gene to future generations.
Anticipating the loss of quite a few rats, we start with 5 couples, 10 rats in total. The amount of offspring will vary with each couple, but the traditional rice rat has about 20 to 25 rats over the span of 6 litters in 1 year. The full experiment is expected to take about a year to allow time for the first group's offspring to mature into adulthood and have offspring of their own. We expect about 3/4 of the litter to acquire the gene from the parent.
Our methods begin with the studying of genetic makeup of various rice rats and to accumulate a number of them that are carriers of HPS. We would begin with a male rice rat that has HPS and mate them with a healthy female rice rat. The male offspring that contains the genetic code that carries the HPS virus will be the main focus. From a 4th male rat, we will take their healthy gene and attempt to replace the HPS gene with this healthy one. They would then mate with a healthy rat who doesn't have the virus.
This next couple, therefore, is the offspring from the first original pair of parents, one that had HPS, one that didn't, and it has been genetically spliced so that the HPS gene has been removed and replaced with a healthy gene. This transformed rat, as it were, is now mating with a healthy rat to see if the gene will pass on to their offspring. Over the span of a year or so, the final litter, born from the genetically modified rat, is expected to have a significant decrease in carriers of the HPS gene. Also, by controlling which gender has the gene in the original couples, we will be able to see who is more likely to pass on the gene, mother or father, based on our results.